second PowerPoint is, I think I called it human viruses. It's a PowerPoint covering a few specific viruses, DNA viruses and RNA viruses that can infect humans. And we'll be using that PowerPoint next week. Just so you know, since next week is the last week before spring break, maybe you guys are almost there, I'm going to reverse the order of the human viruses we discussed. I'm going to start with what I think are the harder viruses, the RNA viruses. So next week, we'll be talking about influenza viruses, HIV, and poliovirus very briefly. And then we're going to switch over to DNA viruses. We'll be talking about herpes viruses, human papillomaviruses, and hepatitis B virus. Right, so we'll do RNA viruses first because their replication cycle is much more complicated, and then we'll talk about the DNA viruses. There's a green multi-page handout here. This is on a couple of the DNA viruses we'll be describing, human papillomavirus and herpes viruses. And just a heads up, you guys, when we talk about the HPV and herpes viruses, some of the strains are sexually transmitted, so some of the slides are going to be showing genitalia that's been infected with these viruses. So I just want to give you the heads up because I know I don't need to offend anybody, but since most of us are going into the health sciences, you being comfortable with the human reproductive tract, I think is important as healthcare professionals. But again, I don't need to offend anybody with this picture. So maybe you want to preview the PowerPoint slides before lecture, and if you find any of them really offensive, what I would always do if like I was feeling queasy in my stomach, you know, I would just look down, like right? Okay. So folks, we're then going to start our investigation of viruses, and this is one of my favorite descriptions of viruses that was made by a virologist, somebody who studies viruses. A journalist was asking this virologist, can you describe to the public what viruses are? And he said, viruses are trouble wrapped in protein. And I just love it. I say, oh, that's a perfect description of a virus. So we're going to explore, you know, what did he mean by this description that viruses are trouble wrapped in protein? So first of all, we'll do a little bit of history. We want to be aware that for many, many generations of humans, viral pathogens were responsible for absolutely horrific outbreaks of infectious disease. For example, smallpox, influenza, these are infectious diseases that have been with humans for many generations caused by viruses. Because viruses are so little, the understanding of their structure, how they replicated, lagged far behind our understanding of how bacterial pathogens replicate, how they infect people. So as early as the 1800s, some scientists had these hypotheses, these educated guesses, that some infectious diseases were caused by microbial agents smaller than bacteria, but nobody knew what they were, right? Some really important work was done in Russia by a Russian scientist, the name of Dmitry Ivanovsky. Ivanovsky was trying to determine what caused the disease of tobacco plants. And tobacco plants are an important economic cash crop, right, because people use them to smoke. And there was a disease of tobacco plants known as tobacco mosaic disease. And we can understand why. If we look at a healthy tobacco plant, the leaf, nice, relatively uniform green, here's a plant infected with tobacco mosaic disease. It's almost like there's these little yellow mosaic tiles set into this deep green background. So nobody knew what caused this particular disease. So Ivanovsky did some experiments to determine if the disease was caused maybe by a bacterial pathogen. And in his experiments, what he did was that he would take a diseased tobacco leaf, he would grind it up, mix it with maybe like saline, and then that gamish, he would filter through these beautiful porcelain filters that had been developed at the Pasteur Institute. And these porcelain filters, their function was to filter out bacteria and anything larger than a bacteria. So these porcelain filters, the pore size, the holes in the porcelain filters were so small that bacteria couldn't fit through the pores. So bacteria would be filtered out, protozoa would be filtered out, fungi would be filtered out. So the idea is that if you took this gamish from the disease plants, which presumably the pathogen would be present, and you filtered it through porcelain filters, it would filter out bacteria and any other cells. The filtrate then would not have any bacteria, any fungi, any protozoa in it, right? So at this time, people thought perhaps bacteria caused this disease. So using the bacteria-free filtrate, Ivanowski inoculated healthy tobacco plants. And his hypothesis was if bacteria were the cause, he predicted that the experimental healthy plants, would you guess, would they get infected or not infected if it was bacteria that caused this disease? Okay, so remember, he took this, he filtered it through a filter that would remove all the bacteria. He took the bacteria-free filtrate, he inoculated healthy plants. If it was a bacterium that caused this disease, would you predict the filtrate would cause the healthy plants to get infected? No, right? There were no bacteria present. But what was the surprising result? The healthy plants got infected. So Ivanowski was like, wow, this means that the pathogen for this disease is smaller than a bacterium. And at that time, people thought bacteria were the smallest pathogens around, right? He didn't know what the heck it was. So he came up with a term, he called it a filterable agent. So the pathogen causing tobacco mosaic disease, he described it's a filterable agent. It'll pass through these filters, it will filter out all bacteria and larger organisms. So you all know in science it's really important that when experiments are done with new discoveries, somebody else has to repeat your work, right, to make sure maybe you weren't fibbing. So just a few years later, a Dutch botanist, Bijering, repeated Ivanowski's experiment. He got the exact same results. And the reason we bring Bijering up is that he gave this pathogen, this filterable agent, he provided a little bit more description. He called the pathogen a filterable virus. So this is the first time, as far as I know, where the term virus was coined. And Bijering used virus because virus in Latin means poison. So he described this contagious living fluid. They didn't know what the viruses were, but they knew that at least in plants they could cause disease. We're going to fast forward then to Europe during World War I. And in World War I, cavalry was still used in battle. And cavalry is when soldiers ride on horseback and they go into charge and they go into battle. And so the cavalry that was stationed outside, I think it was outside Paris, were having an outbreak, an epidemic of diarrheal disease. Now, can you imagine if you're a soldier, part of the cavalry unit, and you're about to go into battle, and you have a horrible case of diarrhea? It might affect your fighting ability, right? And you guys have probably heard this already, that many battles, many wars have been won by infectious diseases or pathogens, more importantly than your man-made weapons. So this is always a huge concern. The health of troops is always really important during wars. So it was really important that the military folks found out what was causing this dysentery, this diarrhea amongst the cavalry folks. There was a Canadian bacteriologist, I don't know French, you guys, so yell at me, Des Harel, I'm guessing. Hopefully I'm not saying it with the proper accent. And so his job was to try to figure out what was causing the diarrhea. So it would be just like if you guys were sent there to discover the reason for the diarrhea. You would collect feces from the men that were sick, and you would plate it on agar, because you're trying to see, can I identify, maybe it's a bacterium, maybe it's salmonella, typhi, causing typhoid, maybe it's Vibrio cholera causing cholera, right? So he plated the samples of diarrhea, trying to see if he could find maybe it was a pathogenic bacterium, right? But as we all know, feces is mostly bacteria, right? Mostly the mass of feces is bacteria. So the challenge sometimes when you're culturing feces is sometimes you have so many bacteria that when they start growing and dividing, their colonies end up touching one another. So instead of having isolated colonies, you have what's called a lawn of bacteria. confluent growth, where the surface of the plate is totally covered in bacteria. It's almost like frosting the surface of the plate with bacteria. So we call that a lawn of bacteria. So in some of de Herrell's plates, he had these lawns of bacteria, right, from the fecal bacteria. But what he noticed is when he held the plates up to the light, he noticed there were holes in the lawn of bacteria. Holes in the lawn. And that, I know it's kind of hard to see here, you guys, but these little purple dots, these would be actually holes in the lawn of bacteria. And he's like, wow, it looks like something's eating the bacteria, right? So he called this particular agent that was killing or eating the bacteria. What do you think he called that agent? Exactly. So for a bacterial eater, something that eats bacteria, the term bacteriophage was used. Phage meaning something that eats something else, eating bacteria. So just so you know, you guys, these bacterial eaters, we now know they're bacterial viruses. Viruses that infect bacteria. Isn't it wild to think that bacteria suffer from viral infectious diseases? I think that's funny. Oh well. Okay, so bacterial virus is a bacteriophage, and just so you're comfortable, folks, a lot of times in microbiology we shorten the name to phage. So in microbiology, if somebody is talking about a phage, you know it's a virus that infects bacteria. It's a virus that infects bacteria. Good. Okay. Now again, de Herrell, he didn't know what these bacteriophages were, but he came up with a basic life cycle where the bacteriophage would attach to a bacterium. Somehow they would kill it. They'd replicate, they'd be released, and then they attach to neighboring bacteria, infect them, kill them, and so we just kind of move outward, killing one batch of bacteria after the other, and that's what was causing these holes to form in the lawn. These holes, again, you guys, we have a fancy term for them. They're called plaques. Plaques. So a plaque is a hole formed in a lawn of bacteria, a hole caused by bacterial viruses in replicating in the bacteria and killing them. We're going to fast forward then to the early 1930s, and now we're back in California at UC Berkeley, where a biochemist by the name of Wendley Stanley was taking purified preps of the tobacco mosaic filterable virus. He was purifying them, and he discovered that they would crystallize. And this had the biochemists, the biologists scratching their heads, because if the filterable viruses, if they were cells, maybe tiny, tiny cells, you should be able to crystallize them. This is a property of chemicals, right? So in the analysis, Stanley believed that these filterable viruses were made of pure protein. They thought they must be just protein, right? And then additional work over in Britain, a little bit further chemical analysis discovered there's a little bit of nucleic acid in these purified preps of these filterable viruses. So by the late 1930s, chemical composition of viruses was starting to get a handle on that. So these filterable viruses were described as being made of nucleoprotein, nucleic acid and protein. Still don't know what they look like, though. Still don't know what they look like. And the reason was most viruses are so small, we cannot see them with a light microscope, right? So now there's always exceptions to the rule, but most viruses are so small, we can't see them using a light microscope. The resolution of a light microscope is about 200 nanometers. And in general, those viruses are smaller than about 200 nanometers. Some are a little bit bigger, but we're going to say for our class that for most viruses, to be able to visualize them, a light microscope isn't powerful enough. To visualize viruses, a more powerful microscope had to be developed, and that is an electron microscope. So we're going to take a look at when the electron microscopes were developed. So now we've gone from World War I, now we're warming up to World War II. And so this made, collaboration between Germany and countries a little bit more challenging. The first electron microscope was developed in Germany in the early 1930s. And then here in the United States, our first electron microscope was developed at the RCA company in 1939. So we had to have the electron microscope before we could visualize most viruses. So there was a great interest in using the light microscope to try to discover the nature, the structure of viruses. And what they did at that time, they had decided that bacteriophage probably were viruses too, right? They would go right through those porcelain filters. And so they used as their first subject for the electron microscopes, they used bacteriophages. And there's a description of when the first electron microscope images were being developed of the bacterial viruses. This group of scientists that were just, literally waiting there with holding their breath, waiting to see these images. And you can imagine, you guys, when this image appeared, I was like, ah! So one of the scientists says, "My God, they've got tails." He must have been from Austria or Switzerland or Germany. "My God, they've got tails." And to me, this is really intriguing. As humans, when we're presented new information, one way we learn it is to try to attach it to something we know already, right? So that's what this scientist was trying to do. Here was something brand new, an image of a virus never seen before, something totally brand new. So he was struggling to find a way to describe it, comparing it to something he knew, right? "My God, they've got tails." So some of the scientists said, yeah, viruses are sperm-like, because sperm have tails. And other scientists just said, yeah, they're kind of like tadpoles or pollywogs, right, because they have tails. Again, trying to make the connection between something totally brand new, almost like, to me, outer space-like, and something that the scientists knew. So at the time, the scientists that were interested in these bacterioviruses, they agreed that they'd focus on one family of bacterioviruses called the T bacteriophage, and T simply means type. So the reason I bring that up, you guys, is later we'll be talking about T-even bacteriophages. It's just one specific family of bacterial viruses. And again, just some of these quotes, you guys, I just love it. So Max Delbruck, who was one of these early researchers, again, being interviewed by a journalist, and being asked to describe what it was like to be involved in the study of viruses virology. So Max Delbruck replied that the study of bacterial viruses or bacteriophage is a fine playground for serious children to ask ambitious questions. And the reason I just throw this in, you guys, and I'll stop after this, is like, our education, it should be joyful. It should, like, kids learn by playing, right? And to me, it makes me very sad that for a lot of you, I know education is not very joyful, and you don't consider it play. It's very stressful because we're asking you to learn so much, and you're working under really stressful conditions. But I hope one day you too will find that education is like a playground. You just love playing and learning new things. And these researchers, they felt that way. It was like a playground to them. All right, just a little bit of vocabulary here. So when you read, you're going to see two different terms referring to viruses. You're going to hear virus or virion. So I want you all to know that I almost always use the term virus. This is kind of the generic term for a virus. So a tiny infectious acellular agent with nucleic acid, genetic information is nucleic acid, surrounded by proteinaceous capsomeres that form a covering called a capsid. So in regular language, this virus is an acellular agent. It consists of nucleic acid protected by a protein coat. Now, the term virion is describing a virus when it's outside of a host cell. So there's two places a virus can be. Inside a host cell where it's actively replicating, or it can be outside a host cell where it's really kind of inert. It can't do anything. It can't replicate itself when it's outside a host cell. So if you hear the term virion, a virion is simply a virus outside of a host cell. I never use this term, you guys. I just use virus for everything. All right, so just so you know. Now here's some general characteristics of viruses that we just want to go over these general properties of viruses. So first of all, folks, viruses, are they cellular or acellular? They're acellular. They're not made of cells. And Dr. Holland came up with a great simple question to ask to decide whether something is cellular or acellular. And her question is, can the agent synthesize a cell membrane? And if the answer is no, it can't synthesize a cell membrane, it is acellular, right? I was like, oh, that's so simple. Okay, so viruses cannot synthesize a cell membrane, thus they're acellular. Now, what we'll find out later, they can steal cell membrane from host cells. They can steal cell membranes from host cells, but they can't make the cell membrane themselves. Viruses lack ribosomes, so which process can't they perform? Protein synthesis, good translation. Viruses don't carry out metabolism. So just an example, you guys, they can't make their own ATP for biosynthesis, so they steal it from the host cell. This is really cool. For genetic information, viruses do use nucleic acid as genetic information, but it turns out some viruses will use DNA as genetic information, and we call them DNA viruses. Other viruses will use RNA as their genetic information, and thus we would call them what kind of viruses? RNA viruses. Okay. With cells, remember you guys, cellular organisms, all cellular organisms use DNA as their genetic information, and all cellular organisms have both DNA and RNA. Remember, RNA is crucial in being able to transcribe and translate that information in DNA into proteins. Viruses do not grow and divide like cells, but rather they're assembled from parts. Like you would put together a car, that's how viruses are assembled inside a cell. And this last term, you guys, I just want to take a little, just expand on this a little bit. Viruses can only make copies of themselves. Viruses can only replicate themselves when they're inside a host cell. And thus, we call them obligate intracellular parasites. But, just a little bit of word work, because this description confuses folks on lecture exams. So let me just do a little concept math. So if we're talking about obligate intracellular parasites, agents that can only replicate when they're inside a host cell, we want to remember that all viruses are obligate intracellular parasites, but there are some cellular organisms who are also, or which are, also obligate intracellular parasites. For example, there are some bacteria are also obligate intracellular parasites. For example, the bacterial pathogen chlamydia can only replicate itself inside a host cell. The bacterial pathogens rickettsia can only grow and divide inside a host cell. Now, the reason I wanted to put this up on the board is, let me give you two examples of exam questions, and the first one is where people stumble a little bit. Okay, so on previous exams, like on a short answer question, I've asked folks provide evidence that supports the statement that viruses are acellular organisms, right? So support could be they can't make cell membranes, they lack ribosomes, they can't carry out metabolism, they have either DNA or RNA, not both. Those are all statements that violate rules for cells, right? So that helps support the statement that viruses are acellular. Where I see very often folks making a little mistake, and I understand it's subtle, some folks make the mistake and say viruses are acellular because they're obligate intracellular parasites. Is that a true statement? No, right? Because we want to remember there's some cellular organisms like the chlamydia and rickettsia that are also obligate intracellular parasites, right? So being an obligate intracellular parasite doesn't automatically make you acellular. Does that make sense? It's kind of subtle there, but I see that a lot. It's a common mistake that folks make. So I just want to make sure you guys, understood that. Good. Now, we're not going to go over this table, but again, when I see like tables or figures that I think might be good study aids, that I throw them up here. So this is just a table comparing viruses to cells, right? And so obviously on the exam, we would emphasize, what we've talked about would be the emphasis. But this is just a nice table comparing some of the different features of viruses and cells. So what we're going to do next is we're going to talk about the general structure of viruses. And like I always do, you guys, we'll do some word work, and then we'll go through the slides that just repeat what I just said on the board. So let's do structure of viruses. And what we'll first focus on are these first two groups, the naked viruses or non-envelope viruses and the envelope viruses. And we'll give a couple of sentences to complex viruses. Okay, so if we're looking at the structure of viruses, we'll start out with the simplest viruses. These are the so-called naked or non-envelope viruses. And what I'll do is I'll just put a squiggle of nucleic acid here. So this will be the nucleic acid, the genetic information. And in viruses, what could the nucleic acid be? DNA or RNA. That's right. So I'm just saying nucleic acid. I'm not saying what it is. Now, it is important for us to know that that DNA and RNA can be quickly broken down in the environment. There's DNases and RNases that can break down DNA and RNA. So we have to protect that nucleic acid. And the way the bacteria protect their nucleic acid, their genetic information, is they cover the nucleic acid in a protein coat or shell. And this protein coat, it has a fancy name. It's called a capsid. It's made up of little protein subunits called capsomeres. And I put kind of a general polygonal shaped capsid up there, you guys. We're going to see that capsids can have different shapes. And the different shape of the viruses help virologists identify the viruses, just like we use the shape of a bacterium. You know, is it a coccus, is it a bacillus, is it a spiral shape to help us identify bacteria? Virologists use the shape of the capsid to help identify viruses. Okay. Now, another thing we want to make note of here, you guys. Remember how we said that viruses are obligate intracellular parasites? They can only replicate inside a host cell. To get their genetic information into a host cell, the first step is the virus has to attach to the surface of a host cell, right? And this is very specific. Attachment occurs when surface proteins, and let me see if we're going to use, I make these fancy, yeah, what the heck, let's make them fancy. Okay, I'm going to put some little protein spikes here. These little protein spikes, we're going to call the viral adhesins. And we're going to put in parentheses are made out of protein. Okay, folks, so help me out. What does adhesive sound like? Does it sound like adhesive tape? And what is adhesive tape? What's the property of adhesive tape? It's sticky, right? Okay. So the first step in a virus infecting a cell, it has to attach to the surface of the cell it's going to invade. Okay. And so at the very tip, so these special proteins that would bind, if this is a host cell, you guys, okay, so this is supposed to be the host cell. It could be the surface of one of our cells. Those adhesins have to bind to complementary surface receptors on the surface of the host cell it's going to invade. Okay, so it makes sense, doesn't it, that the viral adhesins have to be on the outermost layer of the virus, right, so that they can make physical contact with the surface of the host cell they're going to infect. Good. Okay, so our generic naked non-envelope virus. Now the other group of viruses are called envelope viruses.